as I said, it's a real pleasure to be here, and I hope that it's interdisciplinary enough, this talk. So my lab is interested in the fundamental mechanism of how vertebrate cells, tissue cells, migrate. So the ability of tissue cells to move is critical to development, in which mass migrations of cells take place in the embryo, the establishment and maintenance of the vasculature, the immune response, where cells hunt down invading pathogens and heal wounds. And this is an example of the immune response in the tail fin of a zebrafish. So this is the tail fin of a live zebrafish. This is the main vein running down the center of the fin, and this has been stabbed with a tungsten needle. And the cytokines are released from this area and are sensed by white blood cells in the bloodstream that adhere to the vessel wall and transmigrate through the endothelial vessel and make a beeline right for this wound, where they clean up debris and hunt down the invading pathogens. So you can see them getting through the blood vessel wall and heading right for this wound area. Also, these epidermal cells in the skin eventually will migrate in once the debris has been cleared out to heal the wound. So this is an example of cell migration that happens on a daily basis in your life. In addition to the role of cell migration in development and tissue maintenance, it also contributes to disease. Can you see that? Am I going to have the lights down further? Okay. So if cells that shouldn't be migrating begin to migrate, or those that should be migrating stop migrating, then you can have drastic consequences to the organism. So in this example from John Kondilis's lab, this is intravital imaging of a mammary tumor where the tumor cells have been labeled with a green fluorescent protein, and you can see the collagen, the extracellular matrix tissue from the second harmonic that it generates. And what you see actually is individual cancer cells migrating out of this tumor mass actually caught in the act of metastasis. So when tumor cells acquire the ability to leave the tumor and go into other areas of the body, that's when cancer becomes deadly. So what we aim to understand is the fundamental mechanisms of how cells get out of here and go trucking along these collagen fibrils. What are the fundamental molecular mechanisms that make that able to happen? So we can take cells out of tissue and study them undergoing directed migration in tissue culture. This was pioneered in the 70s by Abercrombie and others. And it's been shown through genetics that many of the molecules that are involved in cell migration in actual organisms are the same molecules involved in cell migration in vitro, in this in vitro model system. So as you can see, this is a very dynamic process, and having it happen in a tissue culture dish makes it much more amenable to study, particularly by light microscopy. So what you see here is the typical morphology of a migrating cell, where if it's migrating in this direction, it spreads out this ruffling dynamic area in the direction that it wants to go, and it leaves behind this trailing tail that has to get pulled along as it goes. So you can describe the behavior this way, but the machinations of the molecules inside that are mediating this dynamic process are elusive by this kind of microscopy. So inside the cell, as we all know, are seen in this electron micrograph from the 50s from Keith Porter's lab, they're subcellular organelle systems that are specialized to perform specific jobs. So these include the membrane systems, such as the Golgi apparatus and the endoplasmic reticulum, that compartmentalize protein production, intake, destruction, and secretion. The nucleus, where transcription is controlled, the transcription of genes is controlled. The mitochondria that provide energy to all of these processes. The adhesive organelles that mediate the interaction of the cells with its extracellular environment, including other cells. As well as the cytoskeletal systems that basically provide the primary structural supporting system that determines the shape, stiffness, and physical characteristics of the cell. So in addition to the energy that's required, the primary specialized macromolecular machines that are involved in cell migration are the membrane systems, the adhesions to the extracellular environment, and the cytoskeleton. So in cell migration, it's believed that specific macromolecular machines, these specific organelle systems, contribute to specific steps of the cell migration process, such that, first of all, the cell needs to know where it wants to go. And this involves determining the direction that it wants to go, or establishing a morphological polarity. So where this ruffling end, it wants to go that way, and this end gets pulled along behind it. And this is known to be mediated by the activation of receptors on the cell surface that then talk to the cytoskeleton, the microtubule cytoskeleton inside the cell, polarize that microtubule cytoskeleton so it points the direction that the cell wants to go, and then the cell, now it knows which direction it wants to go, it has to protrude in that direction. So it's established the polarity. Now the front end of the cell has to protrude. This is believed to involve the assembly of actin filaments just subadjacent to the leading edge plasma membrane, as well as the possibility of directed membrane transport to feed this protruding membrane to allow it to expand. Once the cell protrudes, the protrusion has, in order for it to be productive, that protrusion has to adhere to the extracellular environment. This occurs through the engagement of transmembrane receptors called integrins that bind on the outside to the extracellular matrix, and on the inside they bind to actin-myosin contractile structures that are called stress fibers that generate tension along themselves, which generates traction force on the extracellular environment. Then there has to be a regulation of these adhesions such that these ones stay strong and these ones weaken to allow the advancement of the cell to complete the migration cycle. Are we advancing? No, it doesn't look like we're advancing. There we go. We advanced. Okay, it was me who didn't advance the slides. Okay, so over the typical cell biology approach to understanding the problem of cell migration is to grind up the cell into its component parts, these subcellular organelle systems, grind them up, find out what proteins make them up, localize those proteins back in the cell using immunolocalization techniques, solve the structure of the protein, determine who that protein interacts with so that you can develop specific ways, either pharmacological or molecular genetic, so that you can screw that protein up. When you screw that protein up and the cell stops migrating, you say, oh, that protein is important for cell migration. But the problem with that approach is that this doesn't tell us how macromolecular machines animate themselves in space and time, or how they work together as an integrated system to move the cell. And in the end, if we don't know that, we really don't know how protein X functions in this process, and therefore we're not going to know how protein X, if it's mutated, contributes to human disease. So we really need to understand how the macromolecular machines are integrated with each other in space and time in order to understand a fundamental process like directed cell migration. So how do you study space and time in biology at the level of the cell? You use light microscopy. So light microscopy is the key to space and time in living cells. Light microscopy is perturbing to the cells, so you can study live dynamic processes. And with the advent of green fluorescent protein technology, where you can link a fluorescent molecule specifically to the protein that you're interested in, you can study the dynamics of one component or one component, and how does that relate to the dynamics of the cell. And in addition, if every single protein has one fluorophore on it, and you know the fluorescence signal, you know how many proteins there are. So it's quantitative. The problems are that up until recently, you can only look at one protein at a time. And if we want to understand how machines work together to animate a cell, we want to look at a bunch of different proteins at the same time. The other problem with it is the resolution is on the order of a micron scale. So one of these microtubule filaments that you see here is actually 10 times below the resolution limit of the light microscope. So what we're just seeing is the light microscope image of that microtubule and not at the scale that it actually exists at. So we're about an order of magnitude off of being able to see the dynamics of the individual molecules that make up these macromolecular machines. We can't do anything about the limits of the optical limits of light microscopy using simple optics. So to cure this problem, this one system at a time, in the past 10 years, multi-mode microscopy methods have been developed where you can look at multiple colors of fluorescence. So you can label the microtubules yellow, the actin purple, and the nucleus green, and look at how they interact with each other. You can look at the dynamics of the cell by transmitted light microscopy. And this, your ability to look at these multicolor fluorescents has primarily come about through genetic manipulation of the fluorescent proteins. So what do we do about this con number 2? Well, you can do hardware tricks and mathematical tricks that give you maybe a factor of 2 improvement or 3 improvement in this resolution limit. But I want better than that. I want better. So what has been our solution to allow you to see the dynamics of molecules that are smaller than the resolution of the light microscope? And it's totally simple, and it was discovered by accident. So the principle is, the method that we developed was fluorescent speckle microscopy that I developed when I was a postdoc in the lab of Ted Salmon when I was at the University of North Carolina. And so just to give you an example of how it works, this is this is the leading edge of a migrating cell. So the whole cell would be about this big. The nucleus would be about here. So you're just looking at about a 20 by 20 micron window of the front of the cell. This is the cytoskeletal filaments are at such a density that you can't resolve the individual filaments. So this is the fluorescent speckle microscopy image where what we've done in this case is biochemically isolate the actin, chemically link a fluorophore to it, micro-inject those chemically labeled actin subunits back into the cell. They incorporate into this structure, but this is all you see. So what do each one of these speckles represent? So if we take this small region and zoom it up to the scale of this electron micrograph where we can actually see the individual cytoskeletal filaments, what you see is that each one of these speckles is a diffraction limited. So this is the limit of resolution of the light microscope there, about 250 to 300 nanometers, depending on the wavelength. And within that speckle, happens to be more fluorophores linked to the actin cytoskeleton in this region than there are in this region. So what you're seeing now is maybe the average behavior, instead of in the light microscope of thousands and thousands of molecules, you're averaging the behavior of maybe 5 to 10 molecules. So you're getting close to the dynamics of single molecules. And this occurs at a spatial resolution of about, like I say, about 1/4 of a micron. So I like to say that, so here's a movie showing what this looks like when it's animated. So each one of these speckles is moving. That motion represents the motion of the actin material in which that fluorescent actin is incorporated. So I have the actin in red and the microtubules in green. And the intensity fluctuations of each speckle represent the assembly and disassembly of fluorescent actin subunits in and out of that diffraction limited image region into those actin filaments. And the point that I want to make here is that the information density is huge. In each one of these images, there's about 1,000 data points. And so over time, you film these speckle dynamics over time, you're getting on the order of 500,000 to 1 million data points in a 10-minute time-lapse movie where you take the images. Here, I was probably taking images at about 10-second intervals over the course of about 20 minutes. So we've got 500,000 numbers for you. Okay, so the speckle, what's so great about these speckles is that they're quantitative. They act as a local probe of biochemistry and physics in a living cell. So the biochemistry comes in that the speckle appearance and disappearance encodes the assembly or disassembly of that macromolecular ensemble, or the binding and dissociation of molecules to and from some quasi-stable platform, macromolecular platform. The biochemistry also is quantitative in the sense that the rate of change of intensity of each speckle encodes the rate of assembly or disassembly, or the rate of binding and dissociation. So you've got millions of little k-ons and k-offs. The physics comes from the motion. So the motion of the speckles encodes trajectories and velocities, and thermally induced fluctuations. If you track the thermally induced fluctuations of these speckles, it can actually tell you about the viscoelasticity of the material. We have a resolution these days of about 10 milliseconds it takes to get one of these images, and we can track them with sub-pixel fitting positionally down to about 10 nanometers. There's tons and tons of data, as I said, 50,000 data points in a single cell per time point. Then you can do it in multiple wavelengths for multiple proteins, and you can multiply this number times the number of different fluorophores you're looking at. In addition, it's simple. It's totally simple. All you need to do is put less fluorescent protein in, and you get speckles. So it's compatible with any mode of high-resolution microscopy that you can do. Confocal, total internal reflectance fluorescence, two-photon, multispectral. And as I said, we discovered it by accident. We saw speckled microtubules, and we're like, huh, wonder what those speckles are. So, and if you're friends with a computer vision engineer and mathematician, it's amenable to automated computational analysis. And this is my dear friend and close colleague, Gowden Stanuser, at the Scripps Research Institute, who has made this possible. So how do we use this tool to study how the subcellular macromolecular machines work together to mediate a complex process like cell migration? So I'll tell you a story in 3 parts. How the actin-based cytoskeleton builds distinct machines that can both participate in driving cell migration. How the actin-myosin contractile cytoskeleton is integrated with molecules that mediate adhesion to the extracellular environment. And finally, how the protrusive and contractile behaviors associated with cell migration are organized in space by the microtubule cytoskeleton. Okay, so the actin cytoskeleton. Over the past 10 years, a model for how the actin cytoskeleton drives the leading edge protrusion of a migrating cell, a model has come about based on structural biology and biochemistry. And in this model, so by structural biology, we know that actin filaments form this densely packed networks. And on this end of the filament, this is the only ends of the filaments that grow. So the filaments just subadjacent to the plasma membrane grow. The rest are sort of interknitted into this network, and the opposite ends of the filaments are blocked so that they don't disassemble. So the assembly of these filaments, as subunits come onto there, this pushes the plasma membrane forward. And through biochemistry, we have a model. So now we're at the plasma membrane up here, where a signal transduction cascade is started by the signal molecule RAC that activates this structural molecule called the ARP23 complex that binds to the sides of actin filaments and nucleates the assembly of a new actin filament off the side of a pre-existing filament. So that's what gives rise to this geometry of the structure that they form at certain branches. As these filaments grow, they push the plasma membrane forward. They also push the filaments backward. And as the filaments age, they undergo a regulated disassembly. So what this results in is a treadmill of actin that's assembling here with the plasma membrane pushing forward, the actin pushing backward, and eventually disassembling. And it's thought this occurs in a structure in the cell that's known as the lamellipodium. And it's thought that this ARP23-mediated actin polymerization is the essential in the lamellipodium is the essential first step in the cell migration cycle. So the problem is, how do you test this biochemical and structural model in a live cell? So here is unspeckled actin. So this is when you just put a little bit more actin in, and you don't see these quantitative marks all over the cytoskeleton. How do you know, is there a treadmilling actin array at the leading edge of this cell where actin's assembling here and disassembling here and it's pushing the plasma membrane forward? from these types of images. So when you study the actin, when you look at the actin cytoskeleton by speckle microscopy, you can tell where it's assembling, where it's disassembling, and where it's moving. So here we see speckles appearing all along the leading edge, and they undergo this rearward motion away from the leading edge for a couple of microns. Where's my mag bar? This is like 10 microns. There's my mag bar right there. 10 microns. So they undergo this relatively rapid retrograde motion away from the leading edge for a couple of microns. Then there's this other region where they're undergoing this slower retrograde flow, the slower flow away from the plasma membrane. And back here in the cell body, you have these actin filament bundles that seem to be pushing forward. These two flow fields meet in this convergence zone. So when we quantitate this, we can, by tracking the dynamics of these speckles and their intensity fluctuations, here we have a trajectory map where all of these arrows are the trajectory of these actin speckles, and the velocity is encoded in colors. So we have fast motion in red and slow motion in blue. And here we have a kinetic map where the rates of disassembly and assembly are encoded in red and green, where the rate of polymerization, fast, equals bright red, and the fast rate of depolymerization equals bright green. And from performing this type of analysis on many migrating cells, what we find is indeed there is a lamellipodium at the leading edge of the cell, where we have actin assembly concentrated along the leading edge. This is juxtaposed to a band of rapid disassembly behind it. This corresponds to a kinetic region of the cell. This corresponds to a region of the cell where the actin undergoes this rapid retrograde motion. So we do have this treadmilling array. We know from immunolocalization that ARP2/3 and cofilin are concentrated to these regions. But what's this thing back here? We've got this whole other region behind this, and we call this region the lamellum. Again, it's a very flat region, so it's lamellar. But here, the kinetics of actin are different. There's this slow retrograde motion that we could see in the movies. We see that the ARP2/3 and cofilin, those molecules that mediate this treadmilling behavior of actin, are absent from this region, but instead the molecular motor protein myosin and its regulator tropomyosin are concentrated here. Those molecules are absent from this band at the front of the cell. And if we look at the kinetic map, instead of this treadmilling array where we've got assembly juxtaposed to disassembly, we've got these interspersed spots of assembly and disassembly. So this remodeling, this network that's remodeling sort of randomly in space and time. Between these two different actin-based structures, we've got a place where that we can quantitatively map. It's the place where the net actin assembly is 0. So we have rapid assembly, rapid disassembly, and then before we start, we go through this region where there's very little assembly before we get into this lamellum region. And this corresponds spatially to the place of the highest flow speed gradient. So the actin slows down, there's very little actin assembly, and this marks the border between this lamellipodium and lamellum. So the question is, which one's the important one for cell migration? So we map this lamellipodium-lamellum border, and I think in this case it was mapped by the flow speed gradient, the maximum flow speed gradient. And what we see is that when the cell undergoes a productive advance, it's not the lamellipodium. The lamellipodium sort of fluctuates in width over time, but what happens is you get an expansion of this lamellum structure. It looks as if the lamellum expansion may be the critical step for migration. So we wanted to test this hypothesis. So we go to our reductionist friends who make all these tools to knock out the function of specific molecules, and we knock out this one that's supposed to be the king of the lamellipodium, the ARP2/3 complex, in two different ways. And basically the result is that when we knock off the lamellipodium and using our functional criteria, so we can watch the movie first, instead of having these bands of assembly and disassembly, so this is a kinetic map with the polymerization and depolymerization being mapped over time. Instead of having these tight bands of assembly and disassembly right along the leading edge, it's much more spread out. We don't see the bands of polymerization and depolymerization at the end. By looking at the protein localization, we don't have ARP2/3 and cofilin, these treadmilling mediators at the edge of the cell. We also know by a drug test the motion of the actin in this region should be sensitive in the lamellipodium, should be sensitive to this drug. So functionally, we know we're knocking off this lamellipodium, and instead what we have out there is a leading edge lamellum. It has the kinetic signature where we have these interspersed spots of polymerization and depolymerization, these molecular signature with tropomyosin and the myosin motor extending all the way to the edge of the cell instead of being excluded from that region. And the motion of the actin in this region is dependent on myosin, whereas the motion of the actin in the lamellipodium is not. So we knock the lamellipodium off, the cell still migrates. So clearly, mechanisms other than a lamellipodial protrusion driven by this treadmilling array of actin are not required for mediating directed cell migration. So the question that we're interested in answering, since the lamellum seems to be a critical actin-based structure, who's mediating the assembly of actin in this, and how is the advance of the leading edge in this type of structure where we don't have this treadmilling array, how is the advance of the leading edge occurring in the absence of this lamellum advance? So we've got these two systems, this lamellipodium where we've got this treadmilling array of actin on the front of the cell, and this lamellum where we have actin actin motion that's being driven by myosin motor activity. So how is this actomyosin contraction in this lamellum machine, how is this translated through the adhesion, how is this cooperating with the adhesion structures of the cell to communicate with the extracellular environment? You're generating these forces inside the cell, how is that translated to the outside of the cell? So how do you integrate the actin cytoskeleton? How is the actin cytoskeleton integrated with adhesions? So focal adhesions that you can see here stained in green with the paxillin antibody have been recognized as structures that terminate at the ends of these actin filament bundles. So the actin cytoskeleton is in red here. It's known through electron microscopy that these are areas of very close, tight contact of the cell with its extracellular environment. Through biochemistry and genetics, it's been shown that now I think it's on the order of over 100 different proteins assemble into these structures that are involved both in linking them to the extracellular environment, also in activating signals, extracellular matrix-dependent signals to the inside of the cell, mediating communication between the inside and the outside of the cell. And then they hook up eventually to the actin cytoskeleton. So all of these proteins, we don't want to deal with this complexity. We want to break it down into sort of three classes. And what we'll lump them in are proteins that have the capability by biochemistry to bind directly to the actin cytoskeleton, so these actin-binding proteins. Proteins that span the plasma membrane and can bind on the outside to the extracellular matrix, and those are integrins, or the matrix-binding proteins, and everything else in between that can't bind directly to this or this we'll call a focal adhesion core protein. This consists of both scaffolding proteins as well as signaling molecules. So how does this, when you watch the dynamics of the actin cytoskeleton and the formation of these adhesion structures, what you see is this actin treadmill at the front and this slow retrograde flow behind it. But what you'll notice is that these adhesions stay relatively stationary with respect to the frame of reference of the movie, which is the substrate of the extracellular environment, and that actin sort of flows right over them. So how is that? How is the actin affected by its interaction with the adhesion, and how is motion transmitted through these layers of the proteins to generate force on the extracellular environment? So a hypothesis has been developed since people recognized this retrograde flow of the actin cytoskeleton called the molecular clutch hypothesis, where if you have this either polymerization-driven or motor-driven actin retrograde flow at the front of the cell, if you have to anchor that to the substrate through the focal adhesion in order to either allow this actin to to be linked to something to allow the assembly of the new filaments to drive the leading edge forward, or to resist the motion of a myosin motor pulling on those actin filaments. And if you don't engage it, then you just have straight retrograde flow. So the focal adhesion has been hypothesized to be this adjustable clutch that links the motion generated in the actin cytoskeleton to the extracellular environment. So how could you regulate that in this gamish of proteins that are capable of binding to each other, to actin filaments, or to the extracellular environment? Well, you could unhook it here, you could unhook it here, you could unhook it here or here. And with each of these, you would predict a certain correlation of motion of different proteins with the actin cytoskeleton or with the extracellular matrix. So we examined several proteins, actin binding proteins, 3 different actin binding proteins. These two, there's biophysical evidence for them being involved in force transmission. Core proteins, both kinases as well as scaffolding proteins, paxillin and zixin, and one of these transmembrane matrix binding proteins, an alpha V beta 3 integrin. And what we did was correlate the dynamics of 2 speckle fields, basically. So we speckled the actin, we speckled the focal adhesions in 2 different colors. We tracked the flow of within the focal adhesions of the actin and of the focal adhesion proteins. You get speckles in random positions, so we interpolate it on a grid, and then we get pairs of vectors, and basically we just look at the cosine of the angle between them as a direction coupling measure, and sort of the dot product as a measure of the velocity coupling. And this tells you about how hooked they are to each other, okay? So now we're looking at the integrin, which is a transmembrane receptor that binds the extracellular matrix. And you expect that if it's binding the extracellular matrix, it's probably going to be stationary with respect to the substrate, and that's exactly what happens. So we've got speckled adhesions. So this would be like one big adhesion, but it's speckled, so you don't see the whole thing. You see the actin doing its thing. It's treadmilling slowly past the adhesion. When we track both speckles, we see faster actin and very, very slow to non-moving adhesions. Your eyes tend to grab the ones that are moving, but on average, they're not moving. And if we look at the correlation in the motion, in this case, it's the direction correlation. When they move in the same direction, but for the most part, the integrins aren't moving, okay? Now, if we look at one of these proteins, a different place in this sandwich, vinculin, which is an actin-binding protein, interestingly, so this is one focal adhesion. Stop playing. Did it go again? There it goes. We see the actin flowing over it, but we see focal adhesion molecules actually moving through this adhesion plaque. So we can see that they're moving in approximately the same direction. So our direction correlation is lighting up red. So we've got molecules within this adhesion plaque that people think of as a stable macromolecular complex. Some of the molecules are moving in the same direction and close to the same velocity as actin, and others aren't. So we did this on a bunch of molecules, and these are color-coded. So we've got the actin-binding proteins in green. So this is our control. So we've got red actin and green actin, gives us a correlation of 1, which you would expect. Alpha-actinin, which is sort of a promiscuous, ubiquitous actin-binding protein that happens to also be in focal adhesions, it pretty much acts exactly like actin. But the fascinating thing is that this vinculin and talin, these proteins that are implicated in force transmission, actually move in the same direction, but about half the velocity of the actin cytoskeleton. So either they're spending half their time, well, here, let me get to the picture. Either they're spending half their time bound here and half their time bound here, or somehow they're in the middle in some viscous intermediate that's moving half the speed. These core proteins and the integrins tend not to move at all. So they tend to stay together as an ensemble, and this seems to be the slippage interface within the focal adhesion that may we've actually found that it's regulatable. If we activate RAC, we disengage this interface completely. We haven't found yet what engages it completely, and we haven't correlated that engagement yet. Either we're close to doing it with force, but we haven't really correlated it yet with leading edge protrusion, but obviously these are the directions that we want to go. So in the final couple of minutes, what I'm going to tell you, I'm going to summarize a lot of published work to give you an overview of how we try to think about how the microtubule and actin cytoskeleton are integrated with each other to organize this protrusive activity of the lamellipodium and lamellum and this contractile adhesive activity of actin-myosin and the adhesions, how the microtubules may organize that. So the microtubule cytoskeleton is very different than the actin cytoskeleton. The filaments are much sparser. You can see them individually. They're nucleated by a centrosome that tends to hug up against the nucleus of the cell, and microtubules radiate out away from that centrosome. And at the ends of the microtubules, the minus ends, the so-called minus ends in here, tend to be stably attached to this centrosome, where the plus ends undergo this crazy non-equilibrium behavior called dynamic instability, where they assemble and disassemble and switch randomly between assembly and disassembly. So here you can see the cell dynamics by DIC microscopy, and the microtubules are fluorescently labeled. I think we color-coded them in Photoshop. They were probably red or green, but they're pretty this way. And you can see they're undergoing this growing shortening. What does this behavior have to do with the actin cytoskeletal dynamics? It doesn't look much like the actin cytoskeleton dynamics at all. Well, over 30 years ago, Yuri Vasiliev and then Bob Goldman found that if you get rid of these microtubules, so here you have a migrating cell with the microtubules emanating out sort of in the direction of migration. If you treat the cell with a drug that destabilizes the microtubules and makes them all depolymerized, so now we've got diffusible tubulin in there, you lose this polarity. So no longer do you have protrusive activity in the front and contractile activity back here dragging the cell along. Instead, you get protrusion randomly around the cell periphery and contraction randomly organized within the cell. So somehow these microtubules are providing a vector for the placement of protrusive and contractile activity that gets the cell going in the right direction. So this is like a key. You can't just do this anywhere or you're never going to go anywhere. You can't do these activities anywhere or you're never going to get anywhere. So how are the microtubule cytoskeleton and the actin cytoskeleton integrated to promote directed cell migration? And we have two hypotheses for how they could interact with the actin. One is they could stick to them. They could have a structural interaction. Microtubules and actin could be stuck to each other and push each other around. And this would result in an interdependent organization and dynamics. Alternatively, there could be some regulatory integration where microtubules might organize signals that act on the actin, that tell the actin cytoskeleton what to do, because clearly the microtubules are in charge. You get rid of the microtubules, and the actin doesn't know where to go and do its job. So the microtubules could be putting signals in the right place at the right time. Alternatively, there could be some kind of co-regulation of the actin and microtubules. So basically, we've been trying to test these hypotheses over the past 8 years and have evidence that both of these things are going on in cells. So why is it that we've known about microtubules and actin for 50 years, but nobody knows if they stick to each other or not? Well, because they're everywhere. So they're everywhere in a cell. So just by looking at a localization in a cell, since they're both all over the place, how do you know if it's a specific or non-specific interaction? They're also polymers, so in biochemical assays there tends to be trapping and things like that where proteins just stick to microtubules and actin non-specifically because they're this polymer network and they just trap things. So we thought this was a job for this correlational speckle microscopy to see if things were moving at the same trajectories and velocities inside living cells as some kind of evidence for an interaction between the two. And so basically what we see is that microtubules are undergoing the same kind of retrograde motion, anterograde motion as the actin cytoskeleton. So if you watch these microtubules that are parallel to the leading edge, you can see them flowing backwards at the same rate as the actin filaments around them. If you watch these microtubules in the back of the cell, you can see them being pulled forward at the exact same rate as the surrounding actin. Where's my favorite microtubule of all time? Where is it? Where is it? I love this microtubule. There it goes. Look at that one. It's just getting pulled by its nose right up there. And in the middle, you see, I love that microtubule. in the middle, in this zone where the actin undergoes this convergent flow, you see microtubules becoming uncoupled from the actin and undergoing this buckling and bending behavior, and this actually promotes the breakage of the microtubules, as you can see in this next movie. So here, this is the leading edge of the cell. This is undergoing retrograde flow. If we this motion of the actin backwards, if we track these two speckles in this network, we see that the actin speckles are getting closer together over time, indicating that that actin network is contracting. And we see this region of the microtubule that's spanning that contracting network become uncoupled from the actin cytoskeleton, bend, and then buckle. So when this microtubule bends, buckles, and then breaks, this creates a new plus end that undergoes this dynamic instability behavior. And microtubule minus ends tend not to be stable, and so this microtubule eventually will depolymerize completely. So when you break a microtubule, it's a fast way of getting rid of polymer. So you break the microtubule, this guy disassembles, this guy goes on to be a regular old dynamically unstable microtubule. So as a result of this structural integration between the microtubules and actin, we get the same gradient in microtubule assembly states in the cell that we do of the actin cytoskeleton. So this pushing each other around results in a co-dependent organization and dynamics of the two systems. So that in the front of the cell, microtubules are coupled to this actin retrograde flow in the lamellum, and they continue, for some reason, which I'll get to quickly, they continue to grow forward. So as the cell advances, the microtubules are continuously growing forward, while at the same time they're being swept backwards by the actin cytoskeleton, by being hooked to the actin cytoskeleton. This part of the actin cytoskeleton is contracting forward. This breaks microtubules in the center of the cell and results in their depolymerization towards the front of the cell. So what you end up with is assembly here and disassembly back in the center and rear of the cell. So you've got this gradient in microtubule assembly states. Well, why is this important? Because microtubule assembly and disassembly can regulate signal transduction. So what I'm going to tell you about is, very briefly, is how our lab has shown how microtubule assembly regulates this Rho GTPase. And I don't know if you remember in my actin slide, my actin model slide, this is the top of the signal transduction cascade that activates that whole ARP23 mediated treadmilling lamellipodia machine. So microtubules through their assembly dynamics, tell that actin regulatory protein what to do and where to do it. Okay, so Rho GTPase is a member of a GTPase signal transduction protein family called the Rho family. And these GTPases, when they bind GTP and hydrolyze it, this activates their ability to interact with other proteins. In particular, Rho interacts with proteins that polymerize actin bundles and activate the myosin motor, myosin 2, to generate contractile actin bundles. They also activate the growth of focal adhesions, these adhesion structures. RAC, on the other hand, through a couple of kinases and an adapter protein, activate this ARP23 cofilin treadmilling to result in lamellipodial actin polymerization. So what we discovered a long time ago is that actually the growth of microtubules can activate this signal transduction protein RAC. So how did we discover this? Well, I wanted to understand how microtubule growth contributed to the polarity of the cell. So I thought, okay, I'll depolymerize all the microtubules in the cell and with a drug, wash that drug out, and watch the microtubules regrow and watch the cell reestablish polarity. And I thought this was going to be an exercise in statistics where, you know, if the microtubules went this way, did that edge of the cell protrude or retract? But what happened was very interesting. So we got the microtubule in this drug nicotazole. So we wash out the nicotazole, the microtubules regrow out to the periphery, and what you see in the phase contrast movie is the initiation of that growth corresponds to this burst of lamellipodial ruffling around the periphery of the cell. So this, to me, lamellipodial ruffling, I knew was activated by RAC. I said, gosh, it really looks like microtubules are activating RAC. So we showed that we could inhibit this with a RAC inhibitor. We also showed biochemically that when you induce this burst of microtubule polymerization, that this induces a burst of RAC activity in the cell that can then go on to promote the actin polymerization. So what has sort of shaken out of this, as well as studies from others in the field, is that this microtubule growth activates RAC to promote the lamellipodial protrusion. We have some evidence that this could be mediated by membrane transport, actually the motion of moving specific membranes along microtubules to the right place that may carry this signal. Conversely, others in the field have shown that disassembling the microtubules actually activates this other signal that promotes contraction and adhesion. And this may be by the release of a microtubule binding protein that goes to activate that. So when the microtubules are there, this protein's sequestered. You depolymerize the microtubules, this comes out and activates Rho, this GEF-H1. So we know that now that microtubule growth and shortening dynamics somehow can regulate the activity of actin regulatory proteins. So what makes those microtubules still grow to the edge of the cell? This is what I still didn't understand. So those microtubules keep growing. The cell migrates, and the microtubules, they're undergoing a dynamic instability, but on average, they keep undergoing this growth. And we know that because, first of all, the cell goes forward, but second of all, they're being flowed backwards the whole time, and the lamella is always full of microtubules, so it has to keep growing. So we thought, well, maybe since RAC organizes the actin at the front of the cell, maybe it does something to the microtubules too. Maybe there's spatial cooperation with this one signal. So this is an experiment showing we have an activated RAC versus an inhibited RAC. And these are the microtubule dynamics. What you can see, first of all, we know that retrograde flow is activated by the actin cytoskeleton. So we see activation of RAC promotes this retrograde flow. So we know it's doing the right thing to the actin. But also what we see is that the microtubules tend to grow steadily. You see the plus ends that are sort of skating across the front of the cell. But when we look at the speckles along those lattice, we see the speckles are actually flowing rearward, indicating that more subunits have to be constantly adding to the plus end in order to keep those microtubules at the front. In contrast, when we inhibit Rho GTPase, now the microtubules just go like this. They just switch very, very rapidly between growth and shortening and don't exhibit this net growth. We've done our reductionist duty and mapped the signal transduction cascade leading to these this microtubule growth downstream of RAC through this PAC kinase and a tubulin dimer binding protein, OP18, as well as through these interesting proteins, this interesting protein clasp that binds to just to the ends of microtubules and promotes their growth. So to sort of summarize and bring together these different aspects of these different subcellular machines in cell migration, what we can say is that at the leading edge of, the question is how do these interactions between these subcellular systems drive an asymmetry and protrusion at the front of the cell and contraction and deadhesion in the back of the cell that keeps the cell moving in this direction. So we've got a lamellipodium in the front of the cell that's we've shown directly that this is mediated by actin treadmilling, assembly along the leading edge, and disassembly shortly behind it. In addition, the microtubule cytoskeleton steadily grows out into this leading edge of the cell. And behind that, we've got this contractile actin-myosin machine that we call the lamella, where adhesion to the extracellular matrix, the adhesions forming right along here, is engaged to the actin-myosin cytoskeleton. This contraction of the actin-myosin cytoskeleton leads to the breakage and shortening of microtubules in this region. We know that the actin polymerization at the front of the cell is driven by RAC signaling to the ARP2/3 complex and cofillin that mediates the formation of this treadmilling actin machine along the front of the cell, but also RAC signals to microtubules to promote their growth through these other signal transduction cascades. So we've got the co-regulation of microtubules and actin by the same signal transduction cascade, but we also have the microtubules mediating this positive feedback loop, whereas the microtubules continue to grow through some signal, possibly mediated by a transborder membrane, they continue to keep the RAC activation high. So we've got microtubule growth concentrated here that keeps this pink cloud of RAC active here that drives this positive feedback loop. And in turn, the Rho signal transduction cascade talks to the myosin and formins that generate this contractile actin-myosin network that through this slippage interface with vinculin and talin in the adhesions mediates the linkage of actin-myosin to the extracellular environment, the breakage and shortening of microtubules that's driven by this contraction has been shown by others in the field to drive the activation of Rho, which results in a positive feedback loop in this region. So this is just a cartoon hypothesis, but hopefully we can develop models that would actually support this through the numbers that we generate, basically. In general, I couldn't have done this without all the talented postdocs and students in my lab over the years. Currently, well, Ka, she was the one who did the adhesion stuff. Stephanie did the actin stuff. Torsten Whitman, so Ka's now at the University of Indiana with her own lab. Stephanie's now a postdoc. Torsten is a professor at UCSF. So it's been a wonderful pleasure to train these people to love biophysical cell biology, and hopefully they'll make a contribution in this area in the future. Also, I couldn't have done this primarily without my collaborator Galdens Danuser and his team of mathematicians and engineers that developed the image analysis algorithms that allow us to track these very fragile signals, as well as various collaborators, the molecule mashers, I like to call them, that we get all our reagents from that allow us to manipulate specifically distinct aspects of these macromolecular machines. And I want to thank you very much for your attention. How what? What? No. No, so these cells, you know, first of all, we study cells migrating on a glass cover slip so that we can get our, 240 nanometer diffraction limited resolution. And if you start going up into 3D, your point spread function gets ugly. So the cells that we study actually are thinner than the point spread function. That lamella is only about 200 nanometers thick. So we're getting all the information at least out in that thin area of the cell. In addition, the actin cytoskeleton is cortical. So the microtubules are not corticals, but the actin cytoskeleton, if you do a 3D reconstruction of the actin cytoskeleton by light microscopy and look at it from the side, it looks like this. There's no actin in the middle of the cell. It's just around just underneath the plasma membrane. Microtubules, though, they do fill the center of the cell where all the organelles are bound to them. So we do miss a lot of information there. But that's one of the challenges that I'm very interested in. So I'm setting up some microscopy tools to allow us. I don't think we'll be able to do speckles in 3D, but we can do some just quantitative. We could probably track texture, but I don't think we're going to get those rates of assembly, disassembly in 3D. Just optics will kill us, So I think there's sort of variations on it. So, well, first of all, these are tissue cells that we study. So, fibroblasts or epithelial cells. We primarily study epithelial cells. Things like white blood cells, they do a whole different form of cell motility that is much more amoeboid, where they have a sort of poroelastic cytoskeleton, and they squeeze, and the toothpaste oozes out in this area, and then they contract, and so it's a very different kind of motility. But tissue cells like fibroblasts or skin cells or epithelial cells, I think they all have variations on this theme. They might be more lamellipodia dominated or more lamella dominated, but there's definitely around the periphery of cells this treadmilling network, and in the center of the cells there's actin-myosin contractile structures that are linked to the linked to the adhesions, and I think that most of the actin filaments that are born at the edge of the cell get chewed up by this treadmill and don't contribute very much to the actin filaments that make up this machine, this contractile machine. So I think they're largely distinct, and I think that most tissue, all the tissue cell types that I've looked at seem to have it. Some, like a keratocyte, it has more lamellipodia than lamella, and an epithelial cell has more lamella than lamellipodia. So I think that the lamellipodia is probably the thing it's like the nose of the cell. That it's the signal responsive, like John Condilis would say, it's the signal responsive steering wheel, and it might set up a directionality. So we study epithelial cells that are undergoing a wound healing response, and in that system, it's like you grow. You have epithelial cells. Their job in life is to maintain a barrier. So if they hang on to each other for dear life and don't want to let things through. So if you grow a monolayer of epithelial cells in culture and then cut it, all the cells at the edge of the wound freak out and say, oh my God, I need to be touching other neighbors, and they'll start crawling just in the direction that they don't have neighbors. So our cells are automatically polarized. We don't have to provide a chemotactic signal to them. So I have a postdoc who wanted to test the hypothesis, if you have a fibroblast, for example, and you stick a needle of chemotactic agent in front of it, does it need its lamellipodia to go in that direction or not? So he got sidetracked and never did that, though. But I'll get another postdoc someday. So, well, I mean, I know cells electrotax. They'll migrate up an electrical gradient. I don't know whether. I mean, I would imagine that the basis for that is that there's some membrane proteins that have some preference for some ionic state, and once those get activated, it's like a growth factor receptor on the surface. If it gets activated in a gradient, that starts a machine that is this machine that is the actin polymerization machine that's mediated by RAC, blah 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 blah, and it's not necessarily the electrical gradient inside the cell. But I don't know. Well, you can knock intermediate filaments out, and the cell can still migrate. That's a cell in culture that's not subjected to, nasty shear forces or physical insults of other kind. So the fact that intermediate filaments are apolar, everyone's always said, well, they can't contribute to such a polarized process as cell migration. I think that they're absolutely critical to structurally integrating the cell. And so in that respect, like a real cell migrating in a real physically dynamic environment, I'm sure that they play a role. This thing on a flat cover slip in culture, like I said, you can get rid of them and the cell still migrates okay.